Thank you for coming. Today I'll be talking about the science and law of automated DNA evidence interpretation. I'll be doing this by asking some questions first. What is truth? This is an interesting question because you get different answers from scientists and lawyers. I'll then be teaching much of the science, the way we present it to judges and juries, as well as to scientists and lawyers, in the context of two cases. The first case is a fascinating defense case we were involved in. It was one of the hardest cases we ever did. And then afterwards, I'll talk about a prosecution case. We more usually work with prosecutors and police, but we also work with innocence groups, defenders, all sorts of cases. So you'll be hearing about that. So to make it more real, the science will be introduced in the context of cases that I've worked on. Okay, so what is truth? I gave a talk at Duquesne University maybe 12 years ago about forensic science, and one of the deans commented that forensic scientists are very fortunate. We get to work on what is truth, whereas lawyers can only work on justice. That was what he said. He was a lawyer. So what is truth? Well, poets have said beauty is truth and truth is beauty. Well, that's obviously not true because beauty and truth are two different concepts. So that's not truth. There's many kinds of truth that can be addressed through religious texts, but that's usually not admissible in court as evidence. It's a different sort of truth. Newspapers. I don't think many people think newspapers focus on truth. I mean, the facts may be true, but we don't go to the newspaper to understand DNA evidence. I hope not. Maybe you do. You could go to internet. You could Google and try to find out truth there. But it may have little to do with actual forensic evidence. There are authorities. Some people like to believe that truth comes from authority. But again, that doesn't help when you're looking at forensic science and evidence. Here are different authorities, constitutions, books about how government should work. I gave a talk a year ago at a law school, and a law professor began by saying, we all know what truth is. We're all lawyers here. That meant not me. I'm not a lawyer. We know that truth is what a judge decides after listening to two sides of an argument. Well, that's absolute rubbish, of course. I mean, truth is not given to us by judges. Truth is done through science in different ways. Judges make decisions, but the goal is justice, not truth. Philosophers have asked, what is truth? René Descartes, 300 years ago, said, truth is based on what you think. David Hume in England said, truth is based on what you perceive and what you see. Immanuel Kant from Germany had an idea to address both of those, trying to overcome them with his intellectual power. Truth is what you know from different categories. Ludwig Wittgenstein from Austria in the 20th century, considered by many to be the greatest philosopher who ever lived, studying knowledge and truth, sort of said truth is based on, and meaning are based on what people do and their function. This is an old joke. It may not translate well in India, but it's been around a long time. It's not true, it's just a joke. Socrates from Greece, to be is to do. Sartre, to do is to be. Frank Sinatra, dooby-dooby-doo. So what is science? I'll end here because I want to get into some cases. Science is a search for truth based on empirical testing. If you read the history of science, it's basically the only thing that works for creating new knowledge that can move society forward. Nothing, what philosophers, governments, judges, they don't move us forward. You don't get an iPhone from a ruling. Instead, we do empirical testing in science. And this is so obvious to scientists that it's lost on most of society. If you ask non-scientists what science is, they tell you, oh, it's what I learned in high school. That's what science is. But you know that's not what it is. The concept of science is the scientific method. It evolved maybe 300-400 years ago. There's some phenomenon, there's a hypothesis that could explain it that's proposed. What makes science different is that scientists will then go to experimental data. We design experiments, and then we look at the data and we test the hypothesis on data. This whole concept of conducting experiments in order to test hypotheses on data is only a few centuries old. And that's what scientists do. That's what forensic scientists do. That's why we're here. Now, on the other hand, this type of empirical testing is new to man and society for a few centuries. But we see it in nature for 1 billion years. Nature figured this out a long time ago. Viruses adapt by mutation. Vaccines adapt to the viruses adapt to the vaccines. They test each other. We get evolution. And even our immune system is a form of evolution that happens inside of our body as embryos are growing. It's done by testing. That's why the immune system even works, distinguishing self from non-self. All right. That's science. I'm going to say as a working hypothesis, we're focused on truth through testing. That's what we're going to be considering. How do we represent uncertainty? Whenever you do testing, whether it's in nature or in science, you're going to have uncertainty. And there are ways that have been developed to address uncertainty that are also only a few hundred years old. Probability is a field only developed in the 17th century. We can have the probability of some hypothesis. Before we ever look at data, what do we believe? It can be anything. What do we believe that will happen in a cricket match? Based on our belief, who we want to win, and so on. Then we have conditional probability, which is after having seen data and looked at evidence, now what do we think about the hypothesis? That can be calculated. Likelihood is a much newer concept. It was used first by Reverend Thomas Bayes 250 years ago when he developed Bayesian probability, which is the basis of modern computing and statistics. And how do we know anything? How does data change our knowledge? If it's a rigorous theory and we're representing uncertainty, it's based on Bayes' rule. And this is the notion of trying to explain data. That's what a likelihood is. The data are fixed. Try out different explanations. How well do the different hypotheses, the different explanations, explain the data? That's the basis of modern computing and reasoning. This is going to end soon. This is just an introduction for the three of you who like math. But the point is the philosophy of science and what is truth and information are very much connected, and it was only when probability brought it all together that it could even be written down. In American law, we have standards of admissible evidence. Can a jury hear evidence? Well, the other side can challenge, is this reliable evidence? And in science, that's done by asking, has it been tested? Can the method be falsified? Does it have error rates? But it's based on this philosopher's concept, also from Vienna and Austria, on falsifying. If the probability of the data given a hypothesis is 0, you falsified it, then you've said there's no information. In fact, those theories can't even be used in court if the lawyers argue successfully. Thomas Kuhn was a famous philosopher who wrote the book, The Structure of Scientific Revolutions. The idea, how does science progress? It's a fantastic book. I don't know if you read it or study it here, but I've tried to get students to read it. They don't like philosophy very much, but when does science move to a new science? When do we move from human review of DNA data to computers? That's a scientific revolution. He said, it's when we can explain data in a new way. The probability approach is one. Thomas Bayes combined probabilities, priors, before you see evidence, and likelihood, the probability of data given hypotheses, and created really modern science information theory with probability. Here he's looking, what is the probability of some hypothesis after you've looked at the evidence? That's what we want to know. basic statistics is only giving us probability of data given a hypothesis. How do you invert it? How do you turn it around and say what's the probability of a match, the probability of a DNA genotype based on data? It can only be done with Bayes' theorem and a lot of computing. There's no other way to get that answer. And from those ideas of Bayesian probability, in the 20th century, Alan Turing, considered to be the father of computer science, developed the likelihood ratio, where there's only one hypothesis and its alternative, and that ratio of the probability of evidence given a hypothesis divided by the probability of evidence given an alternative. In other words, how well does the hypothesis explain the data relative to the alternative? How well does assuming that these three people are in a DNA mixture explain the evidence that you see relative to two people or unknowns that don't include a defendant? It's that ratio, that change in probability. So those are ways of quantifying information. All right, so let's move on to something more fun. You've suffered enough. Cybernetics, mainly, we get cases from all over the world. We've worked on over 1,000 cases, and we usually just begin by collecting data. We don't charge, and we tell groups, send us some samples, and we'll tell you what we find, see if you're interested. This case, we received over 75 samples, and we just kept analyzing the data for the defense. The lawyer was brilliant. She became a judge last year, and she thought something was very wrong with this case. Somehow the data wasn't analyzed properly. From other things they knew, they really believed this man was innocent. But the accusations were terrible. Manuel Lopez was accused of rape and murder of his girlfriend's toddler son, a 2-year-old. He was facing the death penalty or life in prison. The child was under 3 years old, and there were bruises on his face, his genitals, his rectum. An autopsy showed brain swelling, skull fracture, bruises, asphyxia, unable to breathe. There was a rectal swab from the boy that showed semen, and the swab matched the defendant's DNA. At this point, most lawyers would just give up, accept some kind of plea bargain, and the man would spend the rest of his life in jail. But Judge Kelly Paul thought, and her senior in the case, the picture isn't adding up. And so I'm going to show you the picture. Everything in this case was a DNA mixture. 2, 3, 4 contributors. DNA mixtures, when you have forensic science, you'll begin in any country with simple DNA from one person. Blood on a knife. Just lots of DNA from one person. Pretty soon you'll see mixtures. You try to analyze rape kits. There'll be 2 people. And there'll be some rules you try to follow. It'll turn out they don't work very well, but people will try to interpret the evidence. Then 3 people will be analyzed. What if there's an elimination, like a boyfriend in the rape kit? Now there's 3 people. It starts getting harder, but you're now doing more mixtures. The evidence becomes more mixtures than simple. And when the technology improves, it turns out almost everything is a mixture, because the labs will accept the cases because they can analyze them. So a mixture is DNA from 2 or more people. These same ideas apply to degraded DNA, small amounts of DNA, or the cases that we get, maybe 4 or 5 people in a degraded sample with very little DNA. That's what we tend to look at. So DNA's mixed together. We want to unmix the DNA and figure out who's there. That's the problem. It's mixed together. We want to unmix. I'm going to keep indicating up here, just for context so we don't get lost. Sometimes I'll be switching between human review of data, what people do with their eyes and computers, versus computer review, which I'll show in blue. So you can keep the context. Sometimes I talk too quickly, and you may think I'm talking about computers when I'm really talking about people. So if you look in the top left, that will help. This is human review. These are the problems you're about to hear the history of. When you only have one person and there's two tall peaks that are from the data with a lot of DNA, life's easy. That's what Applied Biosystems developed in their protocols in the 1990s. That's what we were doing in genetics. Well, there was the stutter problem, which we solved in 1995. I won't go into that. But basically, you're looking at tall peaks. But when there's more than one person, or there's 3 or 4 people, you no longer have one allele, a second allele, some random variation from PCR with the signal way above the noise. Now you get more of one person, less of another person, very little of someone else. And PCR variation, PCR is a random process. The data look different every time you amplify. The less DNA there is, when you drop down from 100 cells down to 20 cells or 10 cells, the relative variation becomes more. And it becomes harder for people to work with that. That's not what people handle. So thresholds from simple reference samples were introduced to look at evidence. They work with simple DNA, but this is more of what DNA looks like. They've got lower peaks, like at 15. You've got taller peaks. At 16, you have these peaks come from alleles, but they're not used as data. The reason is the first methods that are used by labs in a country involve applying thresholds the way a machine manufacturer is not an expert in interpreting data. They help you generate very high quality data. Interpreting it is not their expertise. And so you take the thresholds from single source DNA, you apply the threshold, this pattern of tall peaks, lower peaks, all becomes 2 numbers. Whatever's over threshold, a list, 15 and 16, and they're considered to be equal height. Now, looking at the data, you know that isn't true. You can see there's taller peaks, medium peaks, low peaks. To say that all I'm seeing is 15 and 16 and these other peaks, 17 and 18, don't exist, is not the data. I study a lot of science. I don't know any other field except in forensics, forensic DNA, where data is thrown out, altered, changed, not used. In most of science, and certainly in modern computing for the last 30 years with probability, the starting point is the data. You don't change the data, you're not allowed to. Problems occur of inaccurate answers, inconclusives, biases, and so on. So this is human review. We'll be talking about it. Why was human review so popular around the world 20 years ago? Because it's simple. You take the peaks that are left, so those are whatever peaks were over threshold, you look at their allele frequencies, you add them up, you square them, you're done. You can do it on paper, pad of paper in 5 minutes. It's easy. It's completely wrong, but it's easy. And it makes it possible to start looking at mixtures. And this method of adding together allele frequencies and squaring them is called the probability of inclusion. When you have multiple independent loci, you can multiply those numbers together, and that becomes the combined probability of inclusion, or CPI. In some countries, it's called RMNE, random man not excluded. You'll be hearing a lot about CPI. Think of it, it's the statistic that goes with simple interpretation, lists of alleles and thresholds. That's the information number that's used. In America and other countries, you cannot go to court without a match statistic. It's not admissible. You need to have some expression of rarity. But there are problems. This is a study from the US government almost 20 years ago that showed with simple 2-person mixtures, most labs called the results inconclusive. The labs that put a number to it ranged from tens of thousands, a 1 followed by 4 zeros, to hundreds of trillions, a 1 followed by 14 zeros. Clearly there's a problem if most labs think this is inconclusive and the statistic is ranging over 10 orders of magnitude. There's a problem. That's what they were alerting people to. Bias gets introduced. In most of science, you take your data, you put it into a computer, you get some results, and an expert can explain the results. Not with thresholds. Instead, you take the data. A person looks at the data. They apply thresholds. Something's in the list, something's not on the list. They play with the data. Depending on the software, They might adjust dropout values. They might adjust thresholds that can completely change a statistic. Then a person decides, is the defendant there or not? And in most protocols like this with thresholds, only after the analyst has chosen their data and has decided what the answer is, do they calculate a statistic. It's the exact opposite of most of science. NIST introduced, and the FBI introduced something called stochastic thresholds in a paper in 2009, adopted by Swigdam in 2010 for human review. The paper they published had no data in it. There was no validation. It was just cartoons of what a protocol would be. This approach is to say if there's too much variation in the data, there's a lower threshold, the old one, there's a new threshold. They called it the stochastic threshold for randomness. If there's too much randomness, don't use that test. Throw out that one locus. The result was 2/3 of the loci were thrown out with mixtures. Matched statistics of 1,000,000 became matched statistics of 100. The goal was to eliminate all false inclusions. That's what they wanted to happen, but it didn't happen. NIST did another study in 2013 at the expense of far more inconclusives and throwing out most of the information. On the study of 100 different labs on a 3-person mixture, they found only 6 labs properly excluded one of the contributors. The other 25 in the middle called it inconclusive, and 70% of the labs said, this is informative, the person is there, here's a match statistic. Completely wrong. The person was never there. They were falsely including people, and their match statistics, as shown on the bottom, ranged over 3 or 4 orders of magnitude. One of the labs that got the answer correct was using TrueAllele. It was a South Carolina lab. Looking at actual data of human review, I studied the correlation of CPI, which is the threshold statistic, locus by locus, with actual information, the log of the likelihood ratio. We found essentially no correlation. The R-squared value, I think, was less than 0.20. And in this paper, we talked about what's really happening with CPI. It's not measuring rarity. It's not measuring information. It's just a fancy way of counting up how many tests, how many loci at which an analyst thinks somebody's included. Very sophisticated way of just counting. You could have counted the loci. So here we have a method that can't exclude people. It's not giving you information, and it's just putting a number to what a person already knows. That's not the best science. By 2015, once lawyers, even prosecutors began hearing about all this confusion, they started shutting down laboratories. Washington, D.C., Texas. How come the match statistics kept changing? The answer is, to a scientist, is the method of interpretation changed. You go from one threshold with average match statistics of 1,000,000 down to 2 thresholds with average match statistics of 100. Of course there'll be a change. That's very hard to explain to politicians, lawyers, and courts. Easier to shut down the lab and start again, at least in some states. So in summary, manual mixture interpretation suffers from these problems. It's incomplete, data is discarded, not used, thresholds are applied. It's inaccurate. When you get an answer, it's often, most often, inconclusive. The number you get, the CPI value, will disagree with the true information. The method is subjective. The protocols for CPI involve looking at the evidence data, looking at the suspect, and a person looking at the two together. And looking at the answer together with the data can cause problems. It's inoperative. Hundreds of thousands, possibly millions of cases produced, no information is inconclusive. The method is opaque. We don't know what's going on in someone's head as they're thinking and making all these choices and adjusting parameters. And it's biased. It can only include, gives no answer, and most often it's inconclusive and cases aren't closed, at least from the forensic perspective. Okay. Enter the computer. So over 20 years ago, I invented this TrueAllele computer technology. The concept of it is, don't touch the data, like the rest of statistical science. Instead, build models of how the data are formed from the PCR experiment and the DNA genetic analyzer, and explain the data in thousands or tens of thousands different ways, and use that explanation information that's recorded as numbers together with Bayes' theorem to calculate probabilities just from the data. Don't look at the suspect, don't look at anybody. Just work from the data. Make a comparison afterwards. I can tell you a lot more. I'm not going to talk too much about TrueAllele, except to say that it's modern computing technology. It's over 20 years old. As scientists, we took it through 25 versions over 10 years until it worked on all of our test data from all of the labs and far surpassed what people could do to get right answers. You'll be seeing some validation studies as I'm talking for both the scientists and the lawyers. So the result was a system that could use all the data. That's how it was designed. There's no thresholds. There have been at least 42 validation studies, 8 published in peer-reviewed journals. There's no bias from looking at the defendant because the system doesn't know their profile. It's working out the answer for everyone, not just one person. We've reported results in 46 of the 50 states. We've worked on the World Trade Center. Laboratories use it. We've consulted in about 400 agencies, mainly in the US, but also around the world. It's very transparent. You don't have to know what we're thinking. We give people the math. We let them test the software. We give 4 gigabytes of DVD of training videos, papers, decisions, and software so people can test it. In science, the concept that I give you the tool and the data so you can test it yourself is the ultimate in transparency. Lawyers can argue against it because they have to argue, but as scientists, if you're given a method and a tool to test and you're given the data, that's your starting point, not empty words. And it's neutral. It can statistically include people or exclude people. That's how it has helped defendants. The exclusion has helped with our exonerations and Innocence Project work. And the point is we're using information. We began publishing on the method over 20 years ago. The concept is the variation from PCR measures identity. This paper is from the Journal of Forensic Sciences from 2001. Our patents, this method, TrueAllele is so old in terms of its concepts that our patents have long expired. This is not new technology. Here is a listing of the peer-reviewed validation studies. Almost all of them, anything we're first authors on, are posted on our website, cybgen.com, under the publications. I can make anything available to you. And I'll be talking about some of these studies. And I can't tell you everything. That would be a complete course, just telling you what's in these papers. It actually would be an entire semester. But I can give you one key idea from each one as we go through it. So here is in the journal PLOS ONE, which is an open access journal, we published on an information gap in DNA evidence interpretation. This was in 2009. The data came from the federal government, from NIST. They generated it. We interpreted with computers and with people in different ways, made a comparison. What this figure is showing is a very important concept in science and law. There is a straight line linear relationship that lets you predict how much information you're going to get out of a DNA sample. What do I mean? On the x-axis, on a logarithmic scale of picograms, 10, 100, 1,000, is the amount of DNA in one contributor of a mixture. So if you have 5 people in a mixture, this is the amount of DNA for one of those people. The vertical axis, again on a logarithmic scale, is the likelihood ratio. So the scale, as you're seeing, is going 1,000, million, and so on, or 3, 6, 9, 12, the number of zeros after the 1. This log-log relationship is showing that the amount of information as you go from about 1 cell keeps increasing until you hit the full information value, which is 1 over the random match probability, and then it levels off. It's predictable. We used this in our first admissibility hearing in 2009. Scientists and judges like scientific laws, and this has been repeated over and over again. So that's one useful thing from this paper. This was a study we did with the crime lab in 2011. Here, what we're showing on 8 sexual assault cases of 2-person mixtures. In orange, on the bottom of the slide, for each of these 8 cases, from left to right, that's the amount of information. Most of these axes are the log likelihood ratio. So you're getting about 1 million, 1 trillion, or 6, 12, 18. On the y-axis, how many zeros in the number. This cluster is showing that's the amount of information from human review with thresholds. It's about 6 zeros. At this point in time, CPI was giving around 6 zeros or 1 million on almost all cases. Again, it's not measuring information, it's measuring loci. And the height of these 2 peaks are 2 independent computer runs with TrueAllele. So we're seeing first that the numbers are close to each other. And because they're close, that means TrueAllele is reproducible when it's run. And also the numbers are many log units higher. And when we looked at all 8 of these cases, on average, TrueAllele was getting, whenever a person could get a number, TrueAllele's number was about 6 zeros or 1 million times more. This study looked at many things: sensitivity, specificity, reproducibility. It also did a comparison with human review. What you're seeing from left to right are 85 cases sorted by decreasing information. So on the top left is the most informative TrueAllele value, the most informative log likelihood ratio. That's around 24 zeros, or 1 trillion trillion. Those numbers decrease as you move down. The average is 15 zeros, which is about 1 quadrillion. And without going into the details, you can read the paper. I want to tell you about cases. What this showed is that when the computer got information, 70% of the time human review failed completely. The result was inconclusive. These are simpler 2- and 3-person mixtures. You can expect from an excellent lab with good data, excellent analysts, 70% failure of what the computer TrueAllele can find relative to people. This study was 5 unknown contributors. We did it with the California lab. And what it's showing is whether it's high template or low template DNA, 2, 3, 4, 5 contributors, the behavior on this plot of the amount of DNA versus the amount of information is giving the same slope response. Statistically, in an analysis of covariance, it makes no difference how much DNA is there or how many contributors when you're looking at how the DNA of one contributor affects the amount of information. Again, many other things were measured in this paper. I'm just looking for greatest hits in the interest of time. This paper was done with Dr. Nasir Butt of the Cleveland lab. It showed many, many things. Looking at up to 10 unknown contributors. The paper won a minor award as a noteworthy paper for the other findings. But something that's interesting on the left table, at the bottom here, is that even with 10 unknown contributors, it's considered to be impossible with any other technology. What we found was that their lab running their system gave the same answers as our lab running our system. So independently, we're able to get the same answer. I'm not giving a lecture on this paper, so I will move on. Something else that's important for courts and maybe important for India, for how you end up reporting to judges, something we'll be talking about this afternoon in some meetings, is the ability to calculate error rates. A likelihood ratio is not an error rate. It's a ratio of 2 probabilities. It shows the change in information, the change of one probability to another. That's very important. It's not a probability. It's a measure of match strength. It's the change from randomness to evidence. That's what it's measuring. To measure error takes more work. I will not go into it now. This is a half-hour lecture. But The way you do it is if you have an inclusionary statistic like 1 million, you can compare against thousands of samples and estimate an error rate. But what if you could compare against everyone on Earth, or every possible genotype according to its distribution in the population? How can you compare in 1/100th of a second against a trillion reference genotypes? This paper and the associated patents describe how to do that at very high resolution. Once you work out the distribution of match statistics on an evidence item, you can then look at a defendant's match statistic. The area of the curve is telling you the chance of making a mistake, the chance that it's somebody else at that level. We always report that now. I think that will have scope for India. It turns out in this paper, the same method can be used for validations to instantaneously use genotype probabilities, and that can reduce validation time from a year to weeks, because all the calculations are done automatically and pretty instantly. Now I'm going to talk about California versus Lopez. It's been so long since I mentioned this case. Some of you may have felt, even though it was only half an hour, that this has been eons that have gone by listening to all this, especially the lawyers, sorry. But this is reminding you about the case. A young man is accused of the rape and murder of his girlfriend's toddler son, and there's a rectal swab from the boy's, from his bottom that shows semen. It matches the defendant's DNA. What happened? What did the lab find? What did TrueAllele find? What were the puzzles that we were asking? The questions we couldn't solve for half a year trying to understand. And then at the last minute, days before trial, in long discussions, looking at some transcripts, we saw what the answer was. I could tell you what the answer is, but that would be boring and you'd leave. So instead, I'll show you what we found. This is what we showed the jury. I'm stressing this is how we teach juries. We'll typically take 30 to 45 minutes, depending on the jury and the lawyers. We spend a lot of time teaching the lawyers. We have materials for them. We meet with them. And I won't go through this slowly. I can send you actually talks where we go through this more slowly for training lawyers. We begin by explaining DNA biology, the concept of cells. The human body is made of trillions of cells. There are 23 pairs of chromosomes. They come from parents. It's important when you talk about genotypes that there's 2 alleles. Where do they come from? Mother and the father. And how DNA is text. This is something that you all know as scientists and the public generally doesn't know. It's text that can be read by the molecular machinery inside a cell, either in nature or by a scientist. We use metaphors to talk about what is a short tandem repeat. What is an STR marker? The kits have evolved from 4 markers now to about 25 over the last 25 years. What is it? We use the metaphor of an encyclopedia having 23 volumes, 1 for each chromosome. And then we show what a paragraph in one of those volumes might look like to represent an STR locus. Depending on how excited the lawyer gets, sometimes they get very excited and this goes on for a while. We encourage them to move on, but they don't. We can introduce the notion of an allele as the number of times a word, usually 4 letters, A, C, G, T, whatever it is, but root. So everybody in America knows the baseball song. Mothers must sing it to them in the womb. They all know the song, Take Me Out to the Ball Game. So this is very easy for American juries. And the idea is that the variation that occurs in nature that we're trying to measure is in the number of repeats. That word repeat on this slide, the repeated word root, you're seeing 10 times. But when there's variation at an STR locus, it could be repeated 6, 7, 8, 12, 18. Usually there's about 15 different variants of different lengths. So we first show them that because the data is going to involve different lengths. It's good for them to know. We start talking about variation, and then we teach them what is a genotype. Simple genotype. What does it mean? You know what it means. You've got a pair of alleles at an autosomal genetic locus, and that pair of alleles, maternal and paternal, is a genotype at one locus, but by educating the court, that there's about 100 possible allele pairs. That's roughly 15 times 15 divided by 2, because the order doesn't matter. There's a lot of variation that we're measuring. So there's differences between people. And when you look at 10 or 20 or 25 loci, you get to multiply those possibilities together, and there's a trillion genotypes. We use a barcode metaphor, but only 8 billion people. So genotypes are relatively rare. We explain that. We then talk about simple evidence interpretation. For single source DNA, on the left is DNA. We explain the laboratory will extract DNA, it will amplify, and then detect on a genetic analyzer. In the center is data. This is a cartoon. I'm not showing background noise, stutter, relative amplitude. Juries are not really interested in that. We can tell them on cross-exam, but what is the data? It's the sizing data. The molecule with 10 is shorter than the molecule with 12. So we're seeing on a scale of left to right, a 10 allele is detected by the genetic analyzer, the 12 allele is detected, and in a perfect world, nothing else is detected. So obviously one can infer from this. I've run courses, one of my children, when they was in 3rd grade, he said, I've signed you up, you're giving DNA talks to my camp. It's a science camp. I've been giving you the morning. So, but what I found is 3rd graders understand that if I see a 10 and a 12, the genotype's the 10-12. And then you can make a comparison, and the known can be a 10-12 or not a 10-12. Then we talk about DNA mixture interpretation, and the purpose for the jury and the judge is to see it's basically the same thing. Almost nothing's changed. There, on the left, you see there's more DNA present. The laboratory does the same experiment. It extracts the DNA, amplifies with PCR, and then separates and detects on a genetic analyzer. And now, what's the data we're seeing? Well, suppose there's another person here, and now we see, in addition to the 10 and the 12, there's an 11 allele. This suggests more than one person. The inference that we're talking about on the top right arrow is separating the DNA. Suppose, for the purpose of teaching, we know that the first genotype, the genotype of the first contributor at this locus, is a 10-12. This is the kind of question the computer's asking when it's solving the problem, by doing a lot of conditioning on other variables. What's the genotype of the second contributor? Well, we have to explain the 11. What can we explain it with? Not with an 8 or a 9, they're not on the slide. You don't see them in the data. We can explain it by pairing that new 11 allele with a 10, 11, or 12. So in a simple world, you'd have 3 possibilities shown on the top right, a 10, 11, 11, 11, 11, 12, and they have probabilities. It's for one contributor at a locus, just like we saw with the reference sample, but now there's probabilities that are not 100% attached to multiple possibilities. We'll also explain why the 10, 12 may be more probable, because if you add a 10, 12 with an 11, 12, you get 2 doses of 12, so there's more DNA. So we explain it. We adapt to juries, by the way. We don't memorize speeches. Some juries get everything in half an hour. Other juries may take an hour. We look at the jury, see what they're understanding. So does the lawyer. So this is what becomes different. Computers can use all the data. This is what data looks like in this case for one of the 75 evidence items at the BWA locus. The x-axis is showing numbers that, like 166, 171, this is how many base pairs are in the DNA, the length, the number of letters in the DNA paragraph. The numbers on top is the number of repeats. Once you account for the flanking regions, you just count up the number of repeated segments, 15, 16, 17, 18. The y-axis is the relative fluorescent units from genetic analyzer. It's low-level data. It's not up at 1,000. Some of the peaks are under 100. And so what do we see in data like this? First, we see heights of peaks. We don't see thresholded data. We don't see a list that says 15 and 17. Those 2 numbers are high enough. We don't do that. our TrueAllele analyzes the HID file. It tells us what the allele, actually all events are at every base pair, and then it analyzes it. It needs to know the heights of the peaks. It will look at the pattern. It's trying to explain the pattern. And the main thing that it can look at that people cannot look at is the variation in the peaks. When the computer is comparing proposed patterns that explain data, you'll see one in a second, against the data. So it hypothesizes, what if I have certain contributors I compare with the data? It's looking at the variation between what it's proposing as a model and what it's seeing in the data. And from those discrepancies, it can work out the variation. The variation is what you need to calculate an accurate probability. If you think of any probability formula you've seen, it's something like a difference in values over a standard deviation, right? E to the minus 3. You need a baseline. You need variation. So TrueAllele has to measure the variation in the data to get accurate probabilities. In this case, people applied thresholds. They got less information. You'll see how. We'll show how the computer thinks to a jury. We do it by adding together individuals. Suppose, to explain this data, there's a person with a 15-17 genotype. So we have a certain amount of 15-17. You see those blue rectangles. Those are the two alleles. We represent quantity by the height. So maybe we have half as much, shown in orange, of a second genotype, a 16-17. Those start adding up together. And then there's a third person, maybe a 16-18, shown in green. So what's happening? The computer is proposing different amounts of different genotypes. It's going to test its proposal against the data. Here's another one. So maybe a fourth contributor, shown in red, is a 15-18. The computer will try out tens of thousands of different patterns, putting patterns where the peaks are, where they aren't, correct heights, wrong heights. It's searching. It's trying to explain. Good explanations where when you add together the alleles of the genotypes, you are getting the same pattern as the peak heights, will give more probability, more likelihood to those explanations. And most explanations are very bad. They don't put the genotypes where the data are, and they end up with very low likelihoods. There's a large separation between where the explanation is and where the data is. And so that will give lower probabilities. I would love for half of you to go into this and tell you about the math, and it's in papers, and I can do that afterwards, but I don't have time, and half the room wouldn't understand. You can imagine I'd love to talk about that. But let me tell you about the case. When the computer has finished looking at doing these calculations of good explanations, bad explanations, for all 4 contributors at all 15 loci, that's 4 times 15, or 60 genotypes, one for each contributor at each locus. That's what we'll get at the end when we finish the Markov chain Monte Carlo. This is one of those 60 genotypes that the computer writes onto its hard drive and finishes. And this is what a probability genotype looks like. You've seen them before when you look at paternity. The original papers 100 years ago talked in this language, a probability of genotypes. You've seen this when you've looked at genotypes formed from population alleles. They have probability, kinship problems. What may be new to you is seeing probability for evidence genotypes from phenotypic data, like mixtures. But the scientists have seen these concepts before in other contexts. Of the 100 or so possible allele pairs, the allele pairs are shown on the bottom, the data for this contributor has concentrated probability on 8 or 10 possible allele pairs. It's increased the probability, as shown in the blue bars. The y-axis is probability, x-axis is allele pairs. For these allele pairs, it's reduced the probability for other allele pairs relative to a random person. probability is a zero-sum game. In the end, it has to add up to 1. So where is the data constraining the genotypes to put their probability? This, by the way, never saw a comparison reference. It's not seeing the victims. It's not seeing the suspect. It's just separating the data into these probability genotypes. Now we want match information. So far, there's no person involved for a comparison. This is all being done objectively by a computer. The question we want to answer, which is one of about 10 ways of writing down the likelihood ratio with Bayes' theorem, this is the most understandable one. How did the computers looking at the evidence data change the probability from a random person, shown by the brown bars on the bottom, that is, think of allele pair frequencies, to the probabilities we see, shown in blue, after the computer's examining the evidence? How has probability changed? How has it increased? How has it decreased? If we take a look all the way on the left at the 1616, you see that the brown probability before it looked at the data is higher than the 2% afterwards. The probability decreased. This would give a likelihood ratio less than 1 that would point away from a person with a 1616. On the other hand, the 1517 shows a large increase from random to evidence. I don't know, maybe it's tenfold. That would be the likelihood ratio. And when there's little change, if the blue and brown, the population probability and the evidence genotype probability are about the same, then mathematically there's little information because the ratio is 1. The log of 1 is 0. I say to this half of the room, not the lawyers. And so that means 0 information. Still haven't made a comparison. All I'm showing you is now you can choose any reference, any genotype, even if it's not a person who exists, just any genotype, and now we can make a comparison. The computer has the probabilities of all the other genotypes written down. I'm just showing you the most probable ones. So we make a comparison with the victim. It turns out that at this VWA locus, at 1517, the victim happens to have 1517. That's what we didn't know. Now it's the first time, actually it could be done on a different computer or on the database. There are many ways of making the comparison in TrueAllele. Now we're going to take the ratio of the probability of an evidence match, that 48% in blue, divided by the probability of a coincidental match, which is about 16%. This is the easiest way to explain a likelihood ratio to courts. There are many hard ways, like the one I started with at the beginning, with Alan Turing and these strange conditional probabilities. This is the change in probability. That factor of 8, going from 6 to 48, is the match statistic. It's showing the change in probability. That's done at all 15 of these locations. The one we just looked at is on the left. Some numbers, here's the likelihood ratio on the bottom x-axis. Here are the 15 loci listed. Some have large numbers, some have small numbers. The one we looked at just now is in between. And because these loci are genetically independent, we can multiply them together. And that product of those 15 numbers tells us to what extent is the victim in the evidence. And the English that we can say is that a match between the bag knot and the victim, Apollo Torres, I'll look for the smallest number, it's down under Hispanic, third one down, is 5 trillion times more probable than coincidence. So think of what it's saying. It's looking at the ratio of the change in probability. There's no mention of conditional probability. There's no mention of likelihood. There's no concept of that. It's what people understand. How do the probabilities change? By looking at the data. How did the probability of match change? Well, a match between the bag knot and the victim is 5 trillion times more probable than coincidence. That's a starting point for scientists and lawyers to begin to explain. It's just like random match probability, except that the numerator isn't 100%. It's less, like 50% or 40%. Otherwise, because the computer has separated into different contributors, we're only doing one contributor versus one contributor in the end. It's just like a simple single source DNA in concept. We can explain it with pictures and words. There's brother, same concept of how we can calculate his statistic. Now getting more into the case. There was a lot of data in this case. Fortunately, our associate at Cybergenetics, Jennifer Brancamontes, was out there for days going through all 75 evidence items with pictures and statistics for all the references. TrueAllele found 4 or 5 other people's DNA that the crime lab didn't detect. We don't know who they are, but we know there are other people whose DNA was there. And we also compared different methods. I will try to capture that for you for this 1 out of 75 items for everything that was compared in just 2 slides. You can imagine how the jury suffered. So this is brief by comparison. So this is a picture of areas near the knot of a garbage bag found outside the house that contained clothing. And the genotype, or the different genotypes that were separated from this 3 or 4 person mixture, are compared in the left column against 6 known references. We could have used negative match statistics. A negative 3 is 1 in 1,000. That's exclusionary. A negative 6 is 1 in a million. The lawyer felt this was so complex, just indicate that they're not included. Probably a good move, or there would have been too many numbers and heads would have started exploding in the jury box. So I'm not showing you the exclusionary numbers. We weren't asked to show them. So you'll see an X here. So in the first column, we see that these 3 individuals, including the defendant, are not present. We see that the brother is present with 4 zeros after the 1, a number in the tens of thousands, and that the victim, Apollo, is present with a match statistic of a log likelihood ratio of 12, which is a 1 followed by 12 zeros, or in American English, a trillion. What about these other references? These references were found by TrueAllele and Cybergenetics, not by the laboratory. And what we're finding, things that the lab couldn't see with their human review, is that there are matches in the hundreds and thousands, 2 zeros and 3 zeros, against these other 2 individuals. So this is showing all 10 comparisons, the 6 knowns and the 4 unknowns, in TrueAllele for this one evidence item. We then separately compared different labs. Originally, the prosecutors contacted Cybergenetics. Those are the results I just showed you. KRCL is the Kern Regional Crime Laboratory. They're a very good customer of ours. We do new technology with them. They're presenting papers on NGS and new technologies that we work with them on. The crime lab in the other county asked them to use TrueAllele, which usually they don't do that, but they weren't sure what to do. And so the crime lab at the time didn't have error rates in place, so they weren't reporting on all their evidence. I think now they are. But where they did report, their results agreed with ours. This is the state lab in Santa Clara County. SCCL is the Santa Clara County Crime Lab. You'll see they found nothing. Their inclusion methods couldn't say anything, even about the most informative evidence. That's what this case really hinged on, was the different worldview of a primitive method that couldn't find anything versus computer methods. I'm just stressing this here, but our work was done independently by one of our customers, and we got the same results. This is the non-contributor analysis that I described, where we can compare against all possible genotypes of everybody on Earth and calculate error rates. The error rate for this match statistic of 14,000 was 1 in 191,000 people would match as strongly. This is a lovely paper. I still enjoy reading it. I go back to it when I need to understand what I did. or built, or what we're doing, published in Helion in 2018. There's a lot of words and pictures, and for people who like it, there's a lot of math. If you understand random variables and you will like DNA, you will probably love this paper. If you don't know what a random variable is, don't read the math. That's all I can say. But it lets us calculate error rates instantly, which we needed for 75 cases. So let's get into what the lawyers are thinking. There are two different views of this. The prosecution is making the case that the defendant raped and killed a 2-year-old boy who lived in his house. That's what they're arguing. By the way, because they believe their crime lab is excellent, they never wavered from this belief. They didn't know what happened when they lost the case. They thought everybody made a mistake. They weren't listening to the evidence. They were listening to their crime lab, and their crime lab was wrong. The defense is saying, no, there's an abused toddler. It turns out there's a long history of abuse of the mother abusing and beating the child. Many visits to pediatricians, broken bruises. The mother is very abusive. And so there's an abused child who died, and the defendant had nothing to do with his death. We do have the evidence to explain. The prosecution said, look, the defendant's hair is on the clothing bag. On the left, we see the defendant. On the right, we see the evidence. Don't these look the same? Well, DNA analysis, if you look at the red square, showed they may look the same, but they're not. In fact, it's not his hair. Great forensic evidence under a microscope, but the DNA that we could find with TrueAllele is showing it's not him. Lab didn't see that. They just called it inconclusive. Inconclusive can lead to an injustice. This was a puzzle. So there are pathology reports, first in the hospital and then an autopsy. And you think about this puzzle. The first examination, the report said that there's the defendant's semen in the victim's rectum weakly positive in the hospital. And yet shortly thereafter, at the autopsy, it's completely negative. Why? What's going on? How does a positive semen turn into a negative semen? And in the hospital, TrueAllele agreed with the path report. There's a match statistic of 3.81. 3 zeros after 1. It's in the thousands. We agree. His DNA is weakly, it's there. But at the autopsy, he's not there. So the DNA is agreeing with the microscope. TrueAllele's DNA is agreeing with the microscopic findings of the pathologist. We don't know how that happened, but that's a big question for us. Why? That's one of our two big questions. By asking these forensic questions, it led us to what actually happened. There was sperm from the defendant on the victim's penis. Was it there? Well, the crime lab said, we see one cell. Must be there. TrueAllele analysis said, there's nothing there that's worth reporting. There's an exclusionary statistic. With one cell, it might be weak, but he's not there. So this is what happened. The crime laboratory looked at 97 evidence items. They reported 43 matches and one unknown person. We reprocessed 77 of the items that we were asked to using TrueAllele. They're identical data. We reported 138 matches. 138 is maybe 3 times bigger than 43 of what the crime lab found. We discovered 5 unknown people, the one the lab found, and 4 other people. I'm not going to go into this. This is a whole other talk about confirmation bias. There's another 30 slides that go with this. I gave it at the American Academy meeting in a special talk 2 years ago. Not for today. It's just fine. I want to get on with the story. Basically, there was confirmation bias in what the lab was doing. They knew the answer. The computer did not have confirmation bias. It didn't know what anybody was looking for. It was just doing calculations. I'm going to show you some pictures. Each picture is 1/3 of a table, 75 evidence items, 25, 25, and 25. The darker the color, the stronger the match, going all the way from just saying it's an inclusion in gray on the bottom to light blue, that's up to 1,000,000, 6 zeros, darker blue, 1,000,000,000, until you're finally getting to basically single source levels with very dark blue. The first 3 charts are the manual review of the crime lab. The next 3 charts are what TrueAllele found. This is what the crime lab found. They found inclusions to the victim. They didn't report statistics. And they see the defendant. They don't see the mother. This is going to become important. The mother's the primary caregiver, both by history and testimony. She's not there. Next, we see more of the victim in the center. We see the defendant in the red column. There's some more matches against evidence items. The mother is not there. The manual methods are not seeing anything else. Here's the victim. They report some match statistics. And then the crime lab on these last 25 items is reporting inclusions and some match statistics to the defendant. I think that's an unknown. It's finding something, and nothing to the mother. So imagine you look at the sky and all you see are clouds and a few lights. That's what you're seeing. With TrueAllele, the sky lights up with stars. You're seeing what happened. It's looking into the evidence. It's not throwing out. It's not doing anything. It's just telling you what's there. We're seeing on the left match to the brother, which we might expect by cross-matching. We're seeing the defendant. We're seeing more of the defendant than the crime lab does, by a lot. We're seeing more of the victim. We're seeing these other individuals unknown to the crime lab are matching, some of them very strongly. These are the first 25. We don't see the mother. Even TrueAllele isn't seeing the mother, the caretaker. We continue with the next 25 items. We see the victim, strong match statistics. The individuals the crime lab isn't seeing, there are matches to. These are other people. The evidence is almost pointing more, if you're going just on counting matches, to the people we don't know than to the defendant. Still no mother. And it continues with the last 25. There's the victim. Everything's based on match statistics. We see other individuals. We see the defendant in the red column, and no mother. So that's really a question. How can someone be the primary caretaker of a child and there's none of her DNA anywhere from this crime scene on the child, on objects? Nothing. I mean, she says she bathed them, no DNA. Does she not have DNA? Oh, is she a robot? I don't know. We'll get to that. So here's our 2 puzzles. And I'll tell you the bravery of the senior public defender, Kelly Kulik, who became a judge, is she's seeing far more matches to her defendant with TrueAllele than with the lab. And she's not fazed. She's saying, the science, that's the science. Now explain it to me. He's not guilty. We know he's just not guilty. So figure out what happened, because if you don't figure out what happened from all this DNA, he may be executed. Okay. Here's the first puzzle. Where's the mother's DNA? It's nobody's understanding this who's looking at the case. Lots of different people left so much DNA. There are so many matches, but the primary caretaker has no detectable DNA. The rectal DNA conflict. How could the defendant's DNA be found in the initial hospital swab, but not soon after the autopsy? It's supposed to be inside the body. What's going on? 2 different views of the crime. They're looking at the same DNA evidence. They're reaching different conclusions. The prosecution raped and killed the child who lived in his house. The defense, there's an abused toddler. He died. The forensic evidence isn't relevant. The problem with this is each of the lawyers are assuming their side, and their conclusion is what they've assumed. There's no science here. They're just arguing, right? My client is innocent, therefore he's innocent. Your client is guilty, therefore he's guilty. There's no real argument involving forensics. That's what scientists are supposed to do. To see it from their view and the way they presented a trial, the prosecution sees crime, the forensics, the DNA, all these objects. DNA is everywhere. He must be guilty. no explanation, but he must be guilty. The defense sees nothing criminal. This is a quote from the closing argument, which I think was 6 hours, trying to explain everything to the jury. Lots of people's DNA was left in a messy, dirty, spermy house. That's her conclusion. So what does the science tell us? All right, we find some transcripts. I'm asking the defender questions, and I said, so what happened with the mother's DNA? And we come across this transcript, and basically, in this interview with the police, the mother says that she had oral sex with her boyfriend, and she had DNA all over her hands, and she cleaned it. His semen was all over her hands, and she cleaned it up with some baby wipes. Completely new information, right? And then she threw them out. All right. Well, if you take a person whose DNA is not seen at a crime scene, who's touching the baby and everything around continuously, and she has somebody's semen on her hands, and she's touching everything, then 1, you're not going to see her DNA, and 2, you're going to see her boyfriend's DNA on everything. Because it doesn't mean anything. We now know there's massive contamination. Whatever DNA anybody had is completely masked by the DNA from the sperm on the mother's hands, who's touching everything. So, therefore, to the lawyers, I'd say, there's no probative value in the DNA because we now know that it can't tell us anything. The amount of DNA from the defendant's sperm is so much more than all this touch DNA. We're just seeing it everywhere. And we know it's true because we're not seeing the mother's DNA anywhere. And by her testimony, she's touched everything. So she's leaving the defendant's DNA on everything. Not intentionally. Doesn't have a science background, but that's what happened. What about the rectal DNA conflict? Again, a few days before I testified, we're looking through transcripts. This is during COVID, so Kelly's in California, I'm in Pittsburgh, we're having Zoom meetings, we're trying to figure it out. And then we read how the swabbing is done at the hospital. This is an interview with a pathologist. And the description is that, in great detail, I'll summarize it, you can read it, is that the pathologist is taking a swab in sterile water, swabbing the outside of the anal region, and then just going in a little bit to sample the rectal region. And it's called a rectal swab. What's the problem? The problem is language. This is an anal rectal swab. They're first cleaning up an area which, if it's contaminated by the defendant's semen, is outside. But they're reporting it, calling it rectal, as if it's inside. And so this was more complex to explain to the jury. They got it, that the mother transferred the defendant's semen from her hands onto the bottom of the toddler. And that's what they're seeing the first time at the hospital. The first pathologist is swabbing the anus on the outside, cleaning it, getting all the semen from the mother, from the boyfriend, onto the swab. And then you've completely contaminated by cleaning up the rectal swab, but you're not calling it a rectal. You're calling it a rectal swab, but it's really cleaning up the outside area. And you're reporting it as if it's just the inside area, which is leading to the criminal charge of rape and sexual assault. Because the prosecution is thinking the only way that his semen could have entered the child is through some forcible rape. But it's being collected from the outside, transferred from the mother on her hands. And at the autopsy, the first pathologist had already cleaned up the bottom, so there's no semen left. And there's no semen inside either, so now it's negative. But these were the two facts that were very hard to explain, and we could explain them by the testimony of the mother and the pathologists. This is what they did. This is why the surprise went away. First pathologist cleaned up the outside, nothing left for the second pathologist at autopsy. They used the words rectal swab. The jury's ready to execute this man. Horrible crime. But It's just words. So the jury, having listened to this, acquitted the defendant of all charges. And the prosecutor, the head prosecutor, the district attorney, was so surprised by the outcome, they actually eliminated the death penalty in Santa Clara County after this case, because the concept that the evidence might have misled them and they would have executed somebody innocent was pretty horrifying. Ultimately, the mother was convicted of something related to the death. I don't remember. Okay, so the issue is, oh, this is great. Not only do I have another half an hour, but you can't leave. So I'm going to tell you about another case, but there aren't that many more slides. I hope I've left some time for questions. This is more typical. This is a prosecution case. This is a double homicide that we worked on about 10 years ago in Pittsburgh. And it's much more typical. Usually the DNA evidence favors the prosecution in the nature of crime. It sometimes favors the defense or innocence. But this story has a lot of lessons. So we begin by saying, with human review, crime labs don't use all the DNA. They apply thresholds. The data just isn't all used. This is the Wolf sisters' homicide. In 2014, two sisters who came from Iowa moved to the Friendship neighborhood of Pittsburgh, Susan Wolf and Sarah Wolf, and they had a home in East Liberty, and they were found killed. There was no motive, no known motive, nobody knew what happened. That's not typical for Pittsburgh that women are found dead in their homes. It's not a thing. The crime lab looked at, maybe 16 evidence items, but their protocols didn't let them use all the DNA data. They really can't handle complex DNA evidence. They applied thresholds, and what they were able to say for most of the evidence is that they couldn't interpret it. They gave many reasons, and you may have seen language like this if you have it in your report. If you see American reports, you see language like this. There was a hat, no conclusions. This is summarizing many pages of reports. There was a cup, insufficient data. DNA under the fingernails of a victim was contaminated, insufficient data. A gear shift in a car that video footage caught somebody driving away in. Insufficient data. The seat lever cannot be excluded. With a weak match statistic, it doesn't mean much. There was a knit hat that I'll come back to later. Insufficient data. There was a sock, too complex, no conclusions. Do you see language like this in some reports? Or people just don't bother writing it down? In America, you have to write down what you didn't find in your report. So this is what they didn't find. TrueAllele can use all the data. You've seen how it tries to explain the patterns, it measures the variation, it works out probabilities, it tries to explain the data tens of thousands of times at every locus, adding together different genotypes that explain the data well or explain it poorly, better explanations, where the data pattern that you see in front of you of middle, high, low, low at this one locus looks like the explanation pattern of middle, high, low, low. There's the match. All right. The math is, the computer doesn't know about a match. It knows mathematically they're close. It can calculate that the heights of the peaks are close to the heights of the rectangles. And then we get an evidence genotype at each of these. I think it's also 15 loci with 3 contributors, each of the 45 loci. This is what the jury sees. We explain the computer concentrates the probability on these 4 allele pairs out of the 100, and then we can make a comparison with the brown bars, which is from population allele frequencies. What are the probabilities of these genotypes? And then we can get ratios like 50% to 5%. It happens to be that if the defendant had a different genotype, it would have been a different number. If he happened to be a 15-16, like we see on the left, it would have been exclusionary. Coincidence had more probability than evidence match. But when we compared with him, the computer said, well, a 16-16 genotype of this locus, 55% divided by 5%, that's the change in probability, that's the likelihood ratio. Now we look at the match information at these 15 locations, the same chart. This is the likelihood ratio on the x-axis, the 15 loci. Again, when we report, we don't want to, if we can, give the largest or the smallest. We always like talking about typical or average. What's representative? And so here you see that the VWA statistic of around 10 is typical. It's not, over 100. It's not. It's just kind of in the middle. And now we can ask, based on the match statistic, not on what we see, but what the computer has calculated, to what extent is the defendant in the evidence? And we can state to the jury and explain further if asked, we've already shown the picture, that a match, see, I have to tell you, you see here the phrase more probable? It's in the middle of the sentence, right? More probable. It took years to come up with language like this. The purpose of using the word more probable is if you don't put that phrase in there, suddenly you're talking about the probability of a match and people get confused. But if you say something is more probable than something else, then you're talking about probabilities, but you're not hurting the minds of the jurors or the judge. You're saying it's more probable, but you're not using so many words that have math with them that jurors don't want to hear you. You are saying it. It's more probable that there was sunshine this morning than rain. You could expand that into the probability of sunshine today was greater than the probability of rain. You said the same thing. You've conveyed the same idea, but it's easier for people who aren't scientists to hear more probable. It's that kind of being careful with language. But this is reading off the math. It's one of the forms of the likelihood ratio. A match between the fingernails and the defendant is 6 trillion times more probable than coincidence. Okay, that's as simple as we can get it. I don't know if the list of references of articles was distributed. That's one of the 4 articles that I sent out that you might want to read if you're interested in how do you explain the likelihood ratio. I think I wrote it 15 years ago. This is what we found. The crime lab could report 5 DNA mixture matches. Most of them weren't very probative. TrueAllele found 17 matches on the same data. The hat was 65,000 to Alan Wade. The cup, 20,000 to Susan Wolf. The DNA under the fingernails, 6 trillion to the defendant, Alan Wade. DNA under fingernails is very probative. How is DNA from a defendant getting under the fingernails of somebody who's dead? It's very hard to explain in court. If they say they didn't talk, they didn't beat, they didn't do anything, there's the DNA. So that's the probative power of mixtures, is that 2 people's DNA is together someplace it's not supposed to be. Whether it's a sexual assault or a homicide, why is the rapist's DNA in the rape kit? It's not supposed to be there. He said he never met her. How could it be there? It's the most powerful DNA evidence is usually mixtures. It explains what happens. It corroborates, it supports the statements that witnesses make about what happened. Anyway, so we continue. Lots of match statistics. And at the trial, there was so much evidence. I didn't watch the closing argument, but the prosecutor put in time sequence each of the items that we found a DNA match to in front of the jury box. And then I was just told about it. And he walked through the story in time, walking with each piece of evidence inexorably leading to the defendant. That's a good moment for a lawyer. It doesn't affect scientists, but he had so much evidence that he could tell the story supported by bank cards, video cameras, there it was. There was the DNA. And with the older method, he had basically nothing. So he was convicted, found guilty, first-degree murder. But there's another moral to the story, which is that, as you can expect, if a crime lab knows that it cannot interpret certain evidence, in the interest of efficiency, it's probably not even going to test it. If someone brings in a doorknob from a crime scene, we've done doorknobs, they've been used to help convict people. There's 5 or 6 people mixed together on a typical doorknob. If a laboratory is struggling to do 2-person mixtures, they're going to say, no doorknobs. We're not going to analyze those. Handguns typically have 5 or 6 contributors. We get a lot of handguns for especially federal prosecutors. They handle a lot of handgun cases. The crime lab says it's inconclusive, but at least we have the data, we separated it out, and we can help the justice system with those. Hats were something that this crime lab didn't accept, generally. So the detectives knew to not even submit a hat because the crime lab wouldn't analyze it. They also had a backlog of half a year or something, but there's too many people. A hat, criminals share hats. It's a social thing. There's a lot of DNA on their hats. So what actually happened is that Alan Wade was the next door neighbor of the Wolf sisters in Friendship. And there had been a burglary of the Wolf sisters' home 6 weeks before the murder. And at that burglary, a hat was found. Now, at the time, the police didn't submit the hat. They knew the crime lab wouldn't analyze it. And the crime lab wouldn't have analyzed it because they don't analyze hats. But after the murder, 6 weeks later, the district attorney's office said, you're dropping everything else in your lab. We need evidence now from everything. And so they did. Everything was done just on this one case, and they produced all this evidence, most of which they were correct, they couldn't interpret, but TrueAllele can. But ask yourself a question. If DNA could be processed automatically and quickly with each step of the process, taking a few hours to extract, maybe one day using robots to PCR, separate, interpret, and in a few weeks, all DNA evidence had been analyzed by this lab, including the hat from this burglary, they would have known that Alan Wade was burglarizing the house. And the district attorney told me that he felt if that had been known, then the sisters never would have been killed because the murderer would have been detected as a burglar a month before. So that's the danger of not fully analyzing DNA evidence. When it becomes inconclusive, evidence that can be used to prevent crime isn't, and crime continues. Think of serial rapists and so on, serial criminals. So I'll just say in conclusion, I'll show 2 slides. Typically, there's no information from complex mixtures. In America, handguns largely go unanalyzed. They may have 4, 5, 7 people on them. Here's typical result. It's a lot of words that says we can reach no conclusions. So government does whatever it does. There's no conclusions. This case was interesting in that we got it, and it never went to trial. Most of our cases don't go to trial because in America, once there's enough physical evidence, defendants will plead guilty. It's a different story, another lecture. You can have entire symposia on them with lawyers, but that's what happened. So what happened in this case, I was told about this both by the prosecutor and by the defender because I was auditing a law class that he was teaching, and he let me audit the class in part because it helped his client, in part. I'm sure he would have let me audit it anyway. It was on criminal procedure. So TrueAllele took the same DNA evidence from a handgun. Let's see how much time. Oh, that's fine. It's a great story. There's some teenagers driving around town. Shots are exchanged. There's a policeman walking by. He thinks they're shooting at him. Finally, they arrest people. They find a gun. Everybody says they didn't touch it, right? Otherwise, we wouldn't be here doing DNA. Oh, I touched it. No, but they all say they didn't touch it. They unmix. Cybergenetics unmixes this DNA mixture from this inconclusive result. We find that there are 4 contributors. Person A is excluded. Oh, now we have information. The charges are dropped against that person. There's no point in taking this person to trial if the computer has shown he's not on the gun. Okay, that's good. For another contributor, there's a match statistic of about 500,000 to a second person. Well, when he's confronted with the DNA report, he confesses that he handled the gun. Gets a lesser sentence, there's no trial, life goes on. So by having the information, the right people are being connected to the right crimes, and the people who are not connected are not being connected, and it's being resolved with physical evidence. It's not going to court with people arguing about evidence that's not that informative. TrueAllele today. So the math, the algorithms, they're over 25 years old when I developed them. TrueAllele is a computer system as we know it now. We started developing as a Bayesian probability system over 20 years ago. We have 10 laboratories who use it regularly. They've done, I don't know if it's 100,000 cases yet, but certainly many tens of thousands of cases. In cybergenetics consulting work, the laboratories use TrueAllele in their own lab, and other groups come to us. When software doesn't work in a laboratory, usually the prosecutor or the defender will come to cybergenetics, often the police. And we counted it up recently, and we've worked for 400 agencies of all types: police, prosecutors, innocence groups, defenders, so on. The innocence cases are great, but I'm out of time. They're wonderful cases to work on. On our webpage, we describe the 11 exonerations we've done so far from wrongful convictions. There have been 42 validation studies. We've given hundreds of talks. I checked with our office today and asked them to count it up. We've educated over 800 students, mainly forensic science students, about TrueAllele. There have been almost 40 rulings across 5 states in the US, some foreign countries, and federal courts. Cybergenetics has testified in over 100 trials, and we've worked on over 1,000 cases. So I think time is good. A lot of information that you can get from our website or from asking me questions or meeting later on. I appreciate your hospitality. It's been wonderful. I can answer questions now, more later, whatever you'd like. Thank you very much.